Hello, everyone. My name is Dr. Andy Kaufman. I've been talking a lot lately about this viral pandemic, and I've learned some things lately that have helped me develop a theory, and I think I know what is really going on here, so I want to give that information to everyone. I want everyone to know that I am a qualified medical doctor, so I put up some information about my background here. I'm board certified. I practice in psychiatry and forensic psychiatry. I also do natural healing consultation. In the past, I've worked in hematology and oncology. I've held leadership positions in medical school. I ran a medical device startup company. And you can see where I had my education. So I feel that I am qualified to talk about this subject matter, and I hope you'll agree. I just want to thank some people out there who have been really instrumental and inspiring to me. So first of all, Dr. Cowan, who has been outspoken as well on this issue, and he really inspired me and gave me some great ideas that helped me put this whole big picture together. I want to especially thank James True, who has been a great friend, confidant, and collaborator throughout this whole thing, and we're definitely going to do a lot more work together down the road. I really want to thank Richie from Boston, Jason Lindgren, and Crow 777 for giving me the opportunity to get my message out there and for being so supportive and open. It's really just been incredible that I've been able to pursue this mission. I want to thank my kids who have been ignored a lot lately since I've been working so hard trying to figure things out. And I'm so proud of them. They're the only children in my area who I know who are not afraid of getting sick, and they're out playing and having a good time. And then lastly, Dr. Lanka and Dr. Turner Banks, who have done just amazing research and scholarship and have written books and papers that have really helped me figure things out. So thank you to everyone. All right, let's jump right in. So I want to start at the beginning at Ground Zero, Wuhan, China, Hubei province. This is the seafood market there. It's an open air market where the first cases occurred. And as you can see from this picture, there's a bunch of meat and animal carcasses just laid out right on the ground. Here's another photograph from the market where you can see that conditions are somewhat less than sanitary. You have live animals, you have dead animals, you have cut up body parts and fluids that all seem to be cross-contaminating. So this is definitely not the kind of market where I'd be wanting to buy my food from, but certainly there was a lot of traffic there, a very big market. So if what we had is a situation where just about 200 people at first became ill with a mysterious pneumonia type illness at this seafood market, and many of the people were actually employed or made their livelihood at the market. So I want to ask the audience out there, if you learned about this information, what would be your first thought about what most likely might be the problem that caused this mysterious illness? So would it be A, a new genetic disease like cystic fibrosis? Would it be B, a new virus, a new viral illness? Would it be C, autoimmune disease where the body is attacking itself, causing the problem? Or would it simply be bad seafood? And I think most of you would agree that the first thought you would have, given the commonality of the market and the sanitary conditions there, is that bad seafood would be an issue and something certainly to look into. However, the scientists in China did not agree, and in fact, they did not look into this issue at all, as far as I can tell. But instead, they jumped right into the possibility of a virus. So this is a quote from the first paper from the group there that claims to have identified this new coronavirus. And what they said is the disease was determined as viral-induced pneumonia by clinicians according to clinical symptoms, including basically a fever, low white blood cell count. Pulmonary infiltrates just means fluid and congestion in the lungs on a chest x-ray. And the people did not get better after 3 days of antibiotics. So they did think of a bacterial infection first and then went right to the virus. And they note that most of the early cases had contact with the seafood market. So it sounds like they were primed to blame a virus on this illness right away. So how did they actually claim to have proved that a virus caused this illness? So what they did is they took only seven out of the almost 200 initial patients who were sick, and they stuck basically a fiber optic camera, like a long tube, down their windpipe into their lungs, and then they squirted a bunch of fluid in there and mixed it around, and it collected whatever debris or cells or chemicals were in the lungs, and then they sucked it back up. And they did take some other body fluids. They did take blood. They took oral swabs and nasal swabs, but it's the lung fluid where they really found think they found what they were looking for. So when they took this lung fluid out, they did not first try to find a virus in there and separate it out and purify it. But the first thing they did was find and separate some kind of genetic material. Quite an interesting strategy, and what they found was some RNA. But I'll tell you that in our bodies, at any given time, there is some free genetic material circulating around our blood and body fluids. And in addition to that, there are genetic material contained in various types of structures. So there's various types of vesicles, essentially just small little sacs of fluids that sometimes contain genetic material. There's also the normal bacteria that live in our body, including in the lungs, and they have genetic material. So there are quite a number of different sources of genetic material. So when they found this genetic material from the lung fluid, they then determined the sequence of it, which is basically the code of the genetic material. So they determined all of the base pairs and the order of that sequence. And then they rushed to rapidly develop a diagnostic test, which is a qualitative PCR, and I'll discuss that a little bit more in a moment. So in other words, before they really proved anything, they already developed a test. Okay, so why didn't they purify the virus, and how do they know what the source is of that genetic material? So it turns out that I was looking in a related area, and I found this study from last summer. And this study was trying to also develop a diagnostic test, but for lung cancer. And essentially, they used the same exact procedure. So they got the lung fluid in the same way with that bronchoalveolar lavage, and they collected that fluid. And out of that fluid, they isolated genetic material, and they sequenced that and they called it a possible biomarker, and this could be a test for lung cancer. So I thought it was quite interesting and coincidental that the same exact procedure was developing a diagnostic test for lung cancer as was developing a diagnostic test for this pandemic pneumonia. So let's look a little bit more in detail at this test. So this is that RT-PCR stands for, it's a mouthful, but it's reverse transcription polymerase chain reaction. And the RT part refers to that we're using it to amplify RNA rather than DNA. If we were using it to amplify DNA, it would just be called PCR. Okay, now I'll tell you some important things about this test. So the most important thing is it's not actually testing for the virus itself, it's testing for a sequence of RNA. Now, that sequence of RNA may be present in a virus, but it also may be present in other things. And so I'll give you an example, and I think I mentioned this before, but let's say we had a person that we wanted to identify. Okay, so James, I'm going to use you this time. So we want to be able to identify James out of a crowd, and we're going to send him to Yankee Stadium to a baseball game. But we're not going to look for James's face or any other part of his actual body. Instead, we're going to use a surrogate marker just like they're using for this test. So we're going to put a cap on James's head. And now we're not going to use the typical navy blue Yankees hat because there are going to be a million people with that, right? So we got a unique Yankees hat with pinstripes on it. So it's white with blue pinstripes. And we're going to have him put that hat on and then go into the stadium and disperse amongst the crowd. And then we have a team of 10 people that we're going to send out into the stadium looking for that hat. Okay, so we do this procedure, and lo and behold, what happens is we actually find 6 of these hats. Okay, so 6 out of 10 people find someone wearing this hat. So we go and identify if any of those people are James. And it actually turns out that none of them are James, because James doesn't like wearing hats. So once he got in the stadium, he gave his hat to some kid who would have liked it, and that was one of the 6 people that we found. So you see that when you are looking for something that's not exactly the thing you're looking for, but it's something that's associated with that thing, you have to really understand the relationship between what you're looking for and that thing. So in other words, we had to ask James, will you be comfortable wearing this hat the whole time, right? And then we also have to know, did other people buy this hat, or is this a one-of-a-kind hat? Right? So you can see it can be very misleading. We can get a lot of false results by using this type of method. So in order to mitigate that, and by the way, this concept was just drilled into us in medical school, that whenever you're evaluating a new test, that you need to compare it to the gold standard. And that's how you know if it's actually valid. So with this COVID-19 test, there has not been any gold standard test that this has been compared to, because the supposed COVID-19 virus has never been purified and visualized. So in other words, if we were able to take people who are ill with this pandemic pneumonia and take their lung fluid, and from that purify out a virus particle that we could identify, and once we purified that, we could actually take the genetic material right from that particle, so we know that it came where it came from. We know it didn't come from anywhere else, because now we have a pure sample of only these particles. Then we would be able to have a gold standard. And so the way you would test this PCR is you would have a group of sick patients, and then you would have a control group of healthy patients. You would perform the gold standard test, so you'd be able to identify the virus out of each of those patients. And then you would compare the results of the PCR test to that gold standard. And that is really critical, because it would allow you to determine or calculate an error rate, because no test is perfect. It's also very, very important that you have that control group, because that control group would not have the virus present at all, and would also should have a negative PCR test. And that's very important in order to help you calculate these error rates. So one example of a type of error that I think we'd be very concerned about with this test, because we don't want to be mislabeled as being positive for this alleged virus and then risk being quarantined or perhaps even detained. So, we want to know the accuracy. Now, there was a paper that came out where they had to estimate the false positive rate, because you can't calculate it since there's no gold standard to compare it against. And they actually reported an estimated rate of 80% in people without symptoms. So what that means is, if you got tested, let's say you were exposed to somebody who tested positive, or you traveled or something like that, and you want to get tested, or you're asked to get tested, there would be 4 out of 5 times that there was a positive result, there would actually be no illness. So this could be a real big problem. It certainly could vastly overestimate the number of cases, and also could have a lot of consequences for you based on this quarantine situation. So just to talk generally about what the PCR test, because there's actually additional error even beyond what I've described. So what this test does is it's really just an amplification strategy. And the reason this is necessary is because we're kind of looking for a needle in a haystack. We might only find a few copies of this genetic material, and if there's only a few copies, we just can't detect it out of all of the other stuff that's in the fluid. So what this does is it's a reaction that actually replicates the strand of RNA, and it makes a copy. So it makes from 1 to 2. So you would run a cycle of this reaction, and you'd go from, say, 1 copy to 2 copies. Then you would stop the reaction and then start another cycle by adding some more materials, and then you go from 2 to 4 and repeat it again, 4 to 8, and so on and so on. And this is an exponential or a binomial expansion. And if you look at the black curve on the graph here, you'll see that's what that represents. So generally speaking, when you're using this test, you want to carry out approximately between 25 and 35 cycles in order to get enough amplification that you can see what you're looking for. If you go too much beyond that, what happens is you end up amplifying the noise. So it seems to be generally represented that the absolute maximum number of cycles that you could do and still get an accurate result is 45. And that's exactly the number of cycles that is recommended for this COVID-19 PCR test. So it's right at the upper limit. And I'm going to share this quote with you from another article about PCR. It says, what PCR does is to select a genetic sequence and then amplify it enormously. It can accomplish the equivalent of finding a needle in a haystack. It can amplify that needle into a haystack. Like an electronically amplified antenna, PCR greatly amplifies the signal, but also greatly amplifies the noise. Since the amplification is exponential, the slightest error in measurement, the slightest contamination, can result in errors of many orders of magnitude. So this is not a very accurate test, especially when you're pushing the number of cycles to get so much amplification. Just a slight mistake can result in false positives. And I think that's what we've been seeing. So I'm going to change gears now a little bit and talk about something different, something that most of you have never heard of whatsoever, and it's called exosomes. And I'm going to talk about what they are and why they're relevant for this discussion. So exosomes are something that is naturally occurring in the body. So I have this diagram here, and if you look at the top right of the diagram, you'll see that this is a normal cell here outlined, and inside the cell you have these vesicles. And inside of our cells there are a number of different little organs, they call them organelles, and they're generally contained by a membrane. They come in different shapes, but lots of them are in this structure. It's essentially like a spherical blob that has the same membrane as is the outer membrane of the cell, a lipid bilayer membrane, and it can contain various types of chemicals. Now, this is a specific type of vesicle that will end up merging with the cell membrane at the surface under certain conditions and release these exosomes out into the extracellular fluid, and they'll get into the circulation and be distributed around the body. So while they're inside the cell, they're called MVEs, which stands for multivesicular endosomes. So in other words, they have a bunch of exosomes or vesicles inside them. And they release these on a regular basis, just day to day. And there are many things, though, that can induce this process and accelerate it and increase the number of exosomes that it releases outside of the cell. And so these exosomes leave the cell, and you see they have these little squares on them. And these little squares are like a lock. And what they do is they go around the body through the circulation, and they're looking for the right key to fit their lock. And that's called the target cell. And depending on what kind of cell releases them, they might have different keys and different locks, so they're targeted to different parts of the body. And these are mostly thought to involve communication. So communication between one cell and another cell, between one part of the body and another part of the body. And they can have many functions in this communication, which I'll get to momentarily. So here on the left, this is an electron micrograph, or a picture from an electron microscope of exosomes. And you can see here an exosome budding out of the cell. And it's essentially a spherical or circular, this is a cross section, so this is like a slice of the tissue, where on the periphery there are like these kind of globular densities or little dots or circles, right? And inside the cell here where it says MVB, sorry, these are the same as MVEs, that's where all these exosomes are when they're inside the cell before they bud out, okay? And now we have a picture on the right which allegedly shows this COVID-19 virus. And you can see that there are these vesicles budding out of the cell in a circular shape with these globular dots on the periphery. So essentially, it's the same thing. Now, you might think that this looks a little bit fuzzier on the exosomes and it looks a bit sharper in this picture. And I want to tell you that the reason for that is because when you're cutting these thin sections of tissue to make these slides, you're using this device called a microtome. It's like a vibrating razor blade. And it's technically difficult, and sometimes the tissue doesn't cooperate well, or sometimes the person is less skilled, and you might not have a perfect slice. On this COVID-19 slide, this is, I've not seen a better slice than this. It's absolutely perfect. The one on the left is a little bit thicker, and so that's why it looks a bit fuzzier and less sharp, but essentially you're seeing the same exact thing. And I want to take a look at the same comparison when we see both of them inside of the host cell. Okay, so on the bottom left here, this is a COVID-19 cell. And you see right here is supposedly this is the virus particle inside the cell. And once again, you see a circular shape. And inside, you see these round globular aggregates. Okay, now look at the top right. And this here is showing you this is less magnification, and this is actually a nerve cell. And this is the MVE. So these are a bunch of exosomes inside here. And you see a circular structure filled with these globular particles. So once again, same thing, also same size, both about 500 nanometers in diameter. Okay, you see the scale lines here, so you can see for yourself. So now we have a series of 2 comparison photographs under electron microscope of the virus and an exosome outside the cell, and a virus and an MVE inside the cell, and you can see that they are identical in appearance. So let's look at actually some physical parameters of these and continue our comparison. So I've mentioned a little bit that they are roughly the same size when they're inside the cell, same size when they're outside the cell. Now there is some variability here. I've looked at lots of these photographs, and I see in both cases there is some variability in the size. So these numbers are not precise, but in every case they are essentially compatible in their range of sizes. Now also the receptor, now this is a key thing. Now remember I mentioned that lock and key mechanism. Well, actually with the COVID-19 papers, they have discovered that on its surface it has a receptor for ACE2. Now ACE2 is an angiotensin converting enzyme. It's an enzyme in our bodies. One of its functions is that it works with the kidney to regulate blood pressure, and there are blood pressure medications that inhibit this enzyme. But in this paper, what they said is that this receptor is actually how the virus invades the cells. Now, I also was able to find a paper where they've identified exosomes coming from our own body that also use the ACE2 receptor as their lock and key mechanism to find their target cells. So exosomes and COVID-19, the same exact receptor on their surface targeting the same exact cells. Now, these both contain genetic material, and it's both in the form of RNA. No DNA, only RNA. And these structures are both found in the lung fluid. So the lung cancer test that I showed you earlier found exosomes in that fluid, and the lung fluid showed the COVID-19 as well. So you can see that the more we take out this comparison forward, we see that they're essentially the same in every important way. So I happen to be looking in the virology literature, and actually they also think exosomes and viruses are possibly the same thing. So this is Dr. James Hildreth, a very prominent researcher and academic physician in the field of virology and HIV research. He's currently the president and CEO of Meharry Medical College, but he was a full professor at Johns Hopkins, and he wrote this paper with 2 of his colleagues there. And what he said, and I quote, "The virus is fully an exosome in every sense of the word." Now, this was just a great confirmation for what I was already thinking, and I was kind of blown away when I read this in the paper, because this was one of the last papers I looked at to find this after I'd already come to the same conclusion. It really helped validate my opinion. So what is it that causes us to make more of these exosomes and throw them out into our circulation? So it turns out that almost every type of insult to the body would actually cause this process to occur. So toxic substances, and I found several papers looking at this. Some of them looked at environmental toxins, such as heavy metals like arsenic, and organic chemical toxins. Also found evidence about bacterial toxins, and I have a slide that I'll show you in a minute. So there's clearly has a role in the communication or possibly removal of toxic substances that damage our cells. Now, interestingly, psychological stress, including fear, which many, many people around the world are experiencing in a very intense way right now, also causes release of exosomes. So you see how this may cause false positive tests. Cancer, as I mentioned before, lung cancer has many exosomes. Ionizing radiation, infection, injury, in fact, any type of immune response, so whether it be to injury, infection, or another disease. Asthma. Now, many papers just said that exosomes are induced by disease, and they didn't mention anything specific, and they seem to be implying that virtually any type of disease can cause this process. Now, I really wanted to find evidence that EMF would induce exosomes, but unfortunately I could not find a paper on this. But I want to say to all of the people in this field of research on exosomes that this would be an excellent contribution to the literature if you were to look at this issue, if various, especially microwave radiation like 5G, would cause this as well. So I want to talk about the second paper, because this is going to help us merge these two ideas of a virus and an exosome, and are they the same thing? So this is also from the group in China. This one is published in the New England Journal of Medicine, and they had a slightly different protocol, but essentially it was very similar. So what they did is, once again, they did that procedure to get the lung fluid, and then they centrifuged it and just took the fluid. So any cells from the host, like any lung cells that might be in there, or bacterial cells, would be stuck in a pellet in the bottom of the test tube, and they just took the fluid off the top, assuming that if there were viral particles, they'd be in that fluid. But they didn't actually then try to purify the virus out of that fluid. Instead, they mixed it with cells that they took from a person who had lung cancer surgery, and they incubated it with the lung cancer cells. Now, you remember I said earlier, and I showed experimental evidence, that lung cancer cells make exosomes. So when they then purified particles after incubating this fluid with the lung cancer cells and then examined them under the microscope, well, were they looking at viral particles, or were they looking at exosomes? And I have two images below, and one is supposedly the viral particle from this paper, and the other is an exosome. Can you tell the difference? Here is another slide showing the functions of exosomes as they can remove toxins. So in the upper right here, these green cells are actually bacteria. And by the way, this image, all electron micrograph images are always in black and white. If you see an image like this has been colorized after the fact, and they did that to help you identify what is what. So I think this is actually quite a nice approach, and this is a real slide. It's very different from some of the slides that you've seen that are just computer graphics that are made by an artist. So these particles that they colored purple are the toxins released by the bacteria, and if they were allowed to contact the cell membrane, they would actually bore little holes in there, and the cell's contents would leak out and it would die. So what's happening is that the cell put out these yellow exosomes once it realized these toxins were there, and you see they're all budding out of the central area of the cell, and you can see they're basically swallowing up the little toxin particles. And so in this experiment, what they actually did is that they found that when they mixed cells with the bacteria, if the cells put out the exosomes that ate up the toxins, then the cells survived. If the cells were mixed with the bacteria and they did not put out the exosomes, then the cells died. So this was done, in a petri dish, not in a person, but so we can't say for sure, but what this is really telling us is that these exosomes help us clear these toxins so that they don't damage our tissue. So a very important function. I also wanted to take a quick look because there have been some recent reports that people who have been ill are having rapid recoveries with a couple of non-traditional treatments. Like, these are not things that would typically be thought of by the medical establishment to use for a quote-unquote viral illness. But there are certainly reports. There was a doctor in New York City who claimed that he cured several hundred patients with hydroxychloroquine. I think he combined it with zinc. Now, hydroxychloroquine was originally a drug for malaria, but it's used in the United States primarily for diseases like rheumatoid arthritis and lupus. And it is not known how it works in those illnesses. And in my experience, it doesn't really work at all in those illnesses. However, there has been a bunch of research trying to figure out what this drug does. And I read a review paper on this, and I found quite an interesting thing. So there are studies that show that this drug can release lysosomal enzymes. So I'll tell you that the lysosome is basically the garbage dump of the cell. So it's another one of these vesicles or sacs, and it has all these enzymes that basically just chew everything up, similar to what happens in your gut when you're digesting food. And any trash that is not working anymore, right, like proteins that have degraded or DNA that was copied in error or things like that, they get sent to the lysosome for destruction and recycling. So it breaks it down into the basic constituents and then is able to send it back to the factory part of the cell where it could be reused to make new molecules. But in the case of this drug, what it actually does is release these enzymes out of the compartment in the cell and into the cell cytoplasm. And this can actually be quite damaging to the cell itself. However, if the cell has been inundated with some kind of toxic substance, these enzymes could help break that down so that the cell could survive. So I think that may be actually how it's helping these patients. Now, vitamin C is the other one that has been reported to be quite successful. And a lot of people think that vitamin C really works by, quote unquote, boosting the immune system. Now, it does have an effect on some of the immune regulatory function, but I do not feel that that's really how it helps in these conditions. In my opinion, it is the antioxidant properties of this drug and possibly the blood thinning qualities that is what really helps this illness. So when you have a toxic exposure, oftentimes that toxin will cause what's known as oxidative stress or free radicals. And we're all familiar with, that it's recommended to take antioxidants and, eat special kinds of berries and things like that. But not too many people understand what that's really doing. But what it does is it kind of acts like a neutralizing sponge for these free radicals. It just soaks them up and quelches them. And what the free radicals do is they have like a chain reaction. It's like they tag someone and make them a free radical and tag the next person and make it a free radical. And then all these molecules end up falling apart and making a huge mess. And the cells can't function. So the vitamin C stops that process dead in its tracks and reduces that damage and prevents further damage. And in my opinion, that's how it works for this type of acute illness. All right, so I'm going to tie this all together, everything I've told you tonight, into what I think is really going on here. So what I think is happening is that, first of all, as I've reported quite a bit, there are really not pandemic levels of people getting sick and dying from whatever's going on. The numbers are far less than a typical flu season. However, there clearly are some people who are getting sick. What I believe is causing this illness is that there is some kind of insult that occurs that causes the damage, and then there is a reaction to it, which is the production of exosomes. And when we are testing for this supposed virus, we are actually testing for these exosomes, which confirms that there's some kind of unhealthy process going on in the body. Now, I talked about the things that can cause production of the exosomes, and I believe that those represent the cause of the illness. And I think that the possibilities are that there is some kind of poison. Certainly, there's lots of precedent for a pneumonia-type illness coming from inhalation of toxic substances. There could be the stress and fear could be the cause. It could represent a regular flu or pneumonia or even a bad cold, and whatever causes those would be the same cause. And lastly, electromagnetic radiation could certainly be a cause as well. There has been a major proliferation and installation of new 5G infrastructure. And there's certainly evidence known that 5G can have adverse effects on one's health, including damaging DNA. And that evidence actually comes from Los Alamos National Laboratories when they've studied microwave and radiation like they use in the body scanners in the airport. And I think it's quite possible that there is not one toxic exposure, but there could be different causes of this illness in different areas, or perhaps even in the same area between different people, that this may not be a uniform cause of the illness, but it does cause a similar constellation of upper respiratory symptoms. And these symptoms are not extremely specific. All of these illnesses, I could list off probably 20 different illnesses that would cause a cough, possible shortness of breath, a fever, and suppress your white count. So there could be several different causes of this, but I do not see any conclusive evidence whatsoever that there is any virus. And certainly you could see that the exosome and the virus are essentially indistinguishable from each other. So let me just reiterate that. So there is some sort of insult or toxin that you're exposed to, it causes illness, and then that stimulates your own cells to induce and produce and put out these exosomes, because they will send the important signal to your body on how to handle this problem. And part of that is they will actually eat up and, like a sponge, soak up these toxins so that they can be safely removed from your body so that you can recover. So what I'm saying is that COVID-19 actually are exosomes and not a virus that invades your body from the outside. It comes from within your body to help you, and it's a reaction to the real cause of illness. And that's where I'm going to end it right now and take questions. And I just want everyone to know that I have a comprehensive list of references for this talk, and this will certainly be published with this, and I will distribute this slide presentation to all of my collaborators.
Fantastic. Thank you so much, Andy. Is it accurate? I'd like to start with the first question, if that's okay. Is it accurate to say that the day before the Wuhan breakout took place, the day before anyone knew anything was wrong, if you were to send a team into that place, that we would find exosomes inside of every single human there at that location?
Absolutely.
And if we were to do that to the most healthy city in the world that we could find, if we were to go into that place and go into the clean mall where everyone washes their hands, would we find exosomes inside of the bodies of all of those people too? Or a majority of them, let's say?
Yeah, well, so if you looked hard enough, you would find exosomes in every single human being. But in order to make it easier to find, you would have to have a certain level that would be a little bit easier to detect. Okay, because remember I talked about that amplification process. And so if something is really at a super low level, it's harder to amplify. So, but the thing is that you don't have to actually be acutely ill to have an excess of exosomes, because let's say that you're a pretty healthy person, but you decide that you want to treat yourself and you go to a fast food restaurant. And there's a bunch of chemicals and toxins in that fast food restaurant. Well, that is going to cause induction of exosome release. Okay, so just because of that, you're going to now have a higher number, even though you're not acutely ill, and it'd be more likely to be able to be detected on a test. And I would say also, if you had some strong emotional things going on, you may not be physically ill, but you may be in a crisis. You may be experiencing grief or anxiety, or as people are now, fear. And that will also cause an increase in the exosomes, and it'll be much more likely to be detected on a certain type of test.
Fantastic. To follow up on that, if I can, you mentioned the RNA, and as a layman, I'm purposely asking this so you'll correct me, for whatever I'm wrong on. The cell is copying DNA. To copy it, it makes RNA. And during that copying procedure, it's much like an MP3 file in the sense that it's copying as fast as it can. And there's going to be errors that crop up during that copying process, and that those errors could easily trigger, maybe, I don't know if it's easily, but those errors could then trigger that same blue and white striped ball cap. Say it could be found on the awning over the bathroom, or on a lady's purse. She could be having a blue and white striped purse, for example. Is that accurate, or am I off there?
Yeah, no. You're very on the money with looking at this, and there are several things to consider about that. Would you mind just repeating that question again, because I just lost my train of thought? Just the beginning part.
The beginning part mainly was during the cell is copying DNA.
Yes, I remember now. It's about the errors. Yeah. So what I wanted to say was that, so we all know about DNA, right? Everybody, even if you don't know how biology works, you know that we have DNA, and you know that we have genes, and it codes some kind of blueprint for how our body's supposed to work, right? So the DNA is mostly just to like store the code. But when we actually express the genes, what happens is that the DNA, it's called transcription, it gets transcribed and forms RNA. And RNA is a bit easier to work with for certain functions because it's generally in a single strand. So if you were to, when you work with the DNA, even when the DNA makes the RNA, like part of it has to separate from the two strands that are in a helix, open it up so that the enzymes can attach to it to make the RNA. But once you have the RNA, it's much easier because it's just a single strand, so it can then go on, and what happens is it actually codes proteins and makes proteins. Whenever you have either replication of DNA, like DNA making more DNA, like when cells divide, or when you have DNA making RNA, there's always errors that are going to occur. So these errors are just a normal process, because think about it, we're human, we make mistakes, right? We can't ever stop making mistakes. We're always going to make them. And the cells are the same way, and the chemistry is the same way, because we're talking about in our DNA in a human, there are 3 billion base pairs. So in other words, the code is 3 billion symbols long. Okay, so that's a lot of mileage. So of course there are going to be errors in there. And in fact, we have a whole set of machinery in our cells to help catch those errors and correct those errors so that they don't perpetuate and cause us disease and dysfunction. And also, when we're exposed to stress or toxic material or radiation, it can increase the error rate, because those things can affect the chemistry of the replication, and they affect the structure of the water. I mean, water is a really important aspect of this, because the DNA itself, for example, when the water is structured, there's a column of DNA of water that runs inside the helix and is critical for it functioning proper in a chemical way. So if we have some kind of toxin or stress or radiation that disrupts the structure of the water, it can actually perpetuate more errors when there's replication and transcription to make RNA. So that's a really good point. Now, the other point I want to mention is also related to the test being wrong. So when they do this PCR test, they're actually using a probe, which is only a short stretch of the sequence they're looking for, that will attach to the genetic material that you want to find and basically be like the start of adding more bases on and duplicating the sequence exactly. And that probe, so basically the probe is the only thing that will match up with what you're looking for. And some of the probes used in the COVID-19 are only like 20 to 26 base pairs long. So if you think about it, if we have a stretch of 3 billion base pairs, right? I think that somewhere in there, you're going to find every single combination of 26 base pairs. So you're gonna, I mean, it's impossible not to be there. So it would be easy to say that you think this sequence is specific to some virus that you think you found, but it could very well just as easily be a piece of our own DNA that has the same exact sequence.
It's a self-fulfilling prophecy is what it sounds like to me.
Yes, that's certainly one way to look at it.
Thank you. Anyone else have anything? Please speak up.
I have 4 questions. So my first question is, when you say the 45 cycles is the sort of upper limit that's going to start to give you results that aren't necessarily indicative of anything, does that add to the 80% likelihood of false positives, or is that part of the 80% false positives?
Right. Well, I don't think that the team that did that estimation really took into account the method of the PCR when they did that rate. What they were trying to look at is people that tested positive, they could follow them out and see if they got sick. But so I look at it as a separate source of error. Now, certainly it would, if you could accurately measure the error rate, if you had a gold standard comparison, it would be reflected in whatever error rate you calculated, right? But since we only have an estimate and it didn't account for it directly, it could be considered as an additional source of error. But it's really, really difficult to know what the magnitude of that is because we have no comparison. And then we also have a situation where there are different protocols that are run in different laboratories in different countries. So I found a protocol from the World Health Organization that is described 45 cycles. But that doesn't mean that if you go to Taiwan or if you go to Sweden or if you go to Mexico, that they're doing it exactly the same way. It's the same thing with the probes. They list, I think, 7 different probes that you can use. And so you have some choices there as to how you go, and it's just going to be like heterogeneous. And I think a really good example of comparison is the HIV test, which is also quite problematic and has many, over 50 conditions that cause a false positive, including pregnancy. Wow. And so they've done a test where they have one person who has already been documented to be HIV positive, and they take their samples and send it out to like 10 different laboratories and see, do those results agree? And what they find is that they do not agree, that they are sometimes negative, sometimes positive. And they've even done this where they've repeated it over time. So they have that person do it one time, then 6 months later, say, they take samples and send it out again. And even within the lab with the same sample, the result is not consistent over time. So in other words, you take my blood and test it now and it's negative, or it's positive, and then test it in 6 months, it's negative. The same blood sample, same person, right? And I didn't do anything different. So it's impossible to tell right now how much of this error is going on because there's so much panic and urgency to everything. And there's certain things like it's really hard to find information about this test, actually.
Okay, thank you. And there's a couple of the pictures of the exosomes and the virus, and the size, the scale is different. But that you were saying they are actually the same size. It's just the scale is reported in different units, I guess.
Yes, that's
Okay.
it. So there was one where it's reported in one is 500 nanometers, and the other one it's 5 micrometers.
Okay.
Those are actually it's the same exact length. Yeah, they try to make it as confusing as possible by not using standards. So I had a scientific calculator that I was using quite a bit to prepare this lecture.
But yeah, because I don't know those units, so that's good to know. And then, what would you say to people who are saying then this seems transmissible? Are exosomes transmissible, or is this, how would that be happening if it's not sort of our traditional understanding of a virus?
Right. Well, I don't think this is really known. So certainly there are exosomes in our body fluids, right? And if they're in the lung fluid, then when you cough, they're coming out of your body, right? So there is some possibility of this. However, think about the function of the exosomes. The exosomes provide a helpful function. They signal to other parts of your body that there's danger and how to deal with it, right? Or they actually even eat up those toxins. Mm-hmm. So if they were transmitted between people, it could actually perform a symbiotic type of function, a beneficial function, that it could help prepare other people that you might face the same toxic exposure. So you should prepare yourself for that. Now, that has not been, there's no studies that I found that show that. So this is purely my reasoning based on what I do know. I can't say for sure. But I do want to say one other thing about that given the size of these particles, that they are so tiny, there's really no way that you can prevent them from getting out or getting in. Right? So, if you look at the pores of the masks people are wearing, they're way bigger than the size of these particles. Yeah. So they're essentially just going to go right through.
So, yeah, because I just was watching something where the professor was describing it as like if you, saw a group of dolphins getting sick, and would you go just pick one dolphin and start cutting it up and looking for things? Or would you say like, wow, there's probably something in the water. Let's test the water and find out what's in the water. And so that's kind of similar to this, is maybe it's not transmitting, maybe there's a whole section of our world that's got a certain toxin and we're responding to it accordingly. But last question, wasn't 5G deployed in Wuhan just before all of this started affecting people?
Yes, I believe it was. Okay. And, I know that there is a lot of speculation and a lot of people actually with very strong opinions that feeling that 5G is the principal insult that has caused people to be sick. Now, I don't want to say, I think that this is a real possibility, and certainly there is a lot of correlations like that there's an installation and then there's an illness, and I think in Italy you can also find a similar pattern. But I think it's important to know that correlation does not prove causation. Right. And that there's really not quite enough evidence right now to say that with any conclusive opinion on it. But I definitely think that it merits a lot more investigation. And, I am very concerned about this deployment of 5G, that even if it's not the cause of this problem, it's going to be the cause of a lot of problems in the future.
Agreed. That was my last question.
Well, I would also, correct me if I'm wrong, sorry to jump ahead here, but this is a proof that your immune system is doing its job, right? Like if you test positive for exosomes, that's like, yeah, your system is working. Am I wrong about that?
Well, I don't think it proves that your immune system is working, but it definitely proves that your body is reacting to something and trying to function to heal from whatever that insult is. And interestingly, there have been, so remember I talked about that lock and key mechanism. So there have been other studies of exosomes showing that they have the lock for certain immune cells. And they are strongly felt to actually play a role in helping the immune system fix some of these problems. So that's an excellent point, James.
So it sounds like that you've isolated what it is not, but Do we have an idea what it is and what they're supposedly these people are experiencing?
You mean what is the real cause of the illness?
Yes.
Yeah, so that's really, really difficult to say because there aren't really any scientists doing any research experiments looking for another cause. I will point out some interesting things that I heard in addition to the 5G hypothesis, but from Dr. Cowan, who, Lindsay, by the way, he's the one that made that dolphin analogy.
Oh, yeah, cool.
And he was really helped me think about this in this big picture way. But so he mentioned that there was a community, and I think this might be in Northern Africa, but I could definitely be wrong. It was definitely in a developing country where they had just recently made people use biodiesel for all of the vehicles, or they rolled out a big fleet of vehicles using biodiesel. And so he was reasoning that probably, this is like from corn or other GMO crops making this diesel, so that when these vehicles are using this fuel, they're actually putting out tons of glyphosate into the air. And then people would be inhaling this possibly, and then that could be a cause in that area of a respiratory illness. So it's quite possible that it could be multiple different causes in different places, and which also would help account for a slightly different constellation of symptoms that might be segregated in certain areas geographically.
So is this really nothing beyond a flu per se? Like the things that we're seeing, is this just a flu and they're saying it's this other thing? They're just taking all of the flu things that are going on all over the world and saying, oh, it's this one terrible thing and we have to stop it.
Right. Well, that is certainly a possibility. And, that has been my main working hypothesis so far, because if you look at the number of cases and the number of fatalities, you'd see that it doesn't even really rise to the level of the flu. So if they took the real number of flu cases for this year and decided to designate a certain portion of those people to test positive and say that when they're having the flu, it's actually this other really scary virus that is more likely to kill you and that we need to take all these precautions, that everything could certainly be explained by that, because when you have the flu, you're definitely going to put out these exosomes, and so you can have a positive test.